Hi again. So, all right, now I'm going to confront you guys with the classic problem of community ecology. And this is an enduring problem. We love it because it never gets fully answered, which means we can keep working on it. And generations of students and faculty and folks have been studying this problem for a long time. So the question is, how can ecologically similar species share the same habitat? These are all large mammalian herbivore species, and they are all living in our representative savanna ecosystem, Gorongosa National Park. So I'm going to leave that question there and tell you, why is that a paradox? Why should that be a problem? Why shouldn't there be all those species coexisting in the same place? Well, to understand that, you really got to understand the competition coexistence paradox, right? And so that's if you have two species that are competing for food. Here we have buffalo on the left and waterbuck on the right, and they're competing for a single food, which is buffalo grass, Panicum maximum. And buffalo grass is especially efficiently consumed and especially preferred by buffalo. And that makes them the dominant competitor in this situation, okay? And over time, what that can lead to is competitive exclusion, right? The competitively subordinate species, waterbuck in this case, will actually go extinct. Competitive exclusion is a classic phenomenon originally demonstrated with paramecia in laboratory environments, but also potentially a risk in the real world. Now, if you want biodiversity, and I for one want biodiversity, then you need coexistence mechanisms. You need mechanisms that enable ecologically similar species to coexist in the same habitat. And there are a couple classes of coexistence mechanisms, and we don't have time to talk about but one of them. But a big one that we're not going to talk about is predation. And especially the phenomenon of keystone predation. As you guys may have heard of keystone species, that term of keystone derives from this idea that, in this case, the starfish will disproportionately prey on the dominant competitors, which are constantly becoming more abundant because they're dominant competitors. And it'll therefore create room for the subordinate mussel or barnacle species to coexist. Okay? But what we're going to be more focused on, exclusively focused on today, is what we call bottom-up mechanisms, and that involves competition between ecologically similar species and how that leads to what we call niche partitioning, so differentiation in resource use, okay? Now, there's a couple of different ways you can think about niche partitioning. You can think about it in space. So here are 6 different antelope species that are coexisting in a sort of grassy, open woodland savanna with a little swamp in it. And they're occupying particular niches in this habitat. So the reedbuck in the reeds over there, the nyala in the deep woods. And this is a common form of niche partitioning. It's associated with the ecologist Joseph Grinnell. And another classic example you guys might have come across is anolis lizards, which partition the vertical tree canopy. So ungulates, or large hoofed mammals, can do that too. Dietary niche partitioning is another key mechanism that we need to consider. And the way this has been thought about in the case of these big, large mammals here is largely in terms of something called the grazer-browser spectrum. So over here on the left we have zebra. That's a grazer, means it eats predominantly grass. And over there on the right we have a dik-dik, and it eats almost exclusively non-grass or browse. And for those of you guys who don't know what a dik-dik looks like, it's a 5-kilogram antelope. We happen to have a replica skull here. So that's dik-dik, and it's got, if we were to look at the cranial morphology and the teeth of that replica, we'd be able to see adaptations to a browsing lifestyle, or rather we'd be able to see the absence of adaptations to a grazing lifestyle. And then in the middle we have what we call mixed feeders. So this impala might eat a 50-50 mix of both grasses and browse. Okay? So that's the grazer-browser spectrum. Another form of dietary niche partitioning is that animals can separate out by height. All right? So this one should make intuitive sense. If you've got 3 large browsers, again, browsers being the ones that eat trees and don't eat grasses. So here we have giraffe, kudu, and steinbuck, and feeding along a height axis on a tree. And the proportion of time that they spend feeding is there on the x-axis, and you see the giraffe spends most of its time feeding on the tall parts of the tree, the kudu at the intermediate parts, and the steinbuck at the lowest parts. Believe it or not, something similar is true for how grazers partition their grass. So if we have, again, our one grass species, this is our Panicum maximum buffalo grass. This is examples from the Serengeti, where they have a long dry season. And when the rains come, the grass grows tall. And the first herbivores to move in to start foraging on that tall grass are zebra, which they're non-ruminant and large body-sized animals, which means they have to eat a lot of food and they don't digest it very thoroughly. So that's kind of their strategy. So they move in and they eat this grass, which is relatively low quality, has a lot of stems. They're followed by wildebeests, who prefer grass of a slightly intermediate height and slightly higher quality, because as the grass gets shorter, then we're getting to the more nutritious parts of it. And lastly, the smallest and most selective herbivore species, Thompson's gazelle, is feeding at the smallest, the shortest grass that you find sort of 6 months after the rain, after the other herbivores have passed through. And this is a phenomenon known as grazing succession, and it also relates to a concept known as facilitation, because zebra are thought to be sort of preparing, if you will, the range or the pasture for wildebeest, and wildebeest further prepare it for Thompson's gazelle. So they're actually a positive as opposed to a competitive interaction among these species in this situation.
African savannas are famous for their diversity of large mammals. But how do so many similar species coexist in the same place? Princeton University biologist Rob Pringle is captivated by this question, one of the enduring mysteries of ecology. His team is working in Mozambique's Gorongosa National Park. Their focus is 3 closely related species of antelope: bushbuck, nyala, and kudu.
They all occur here in the same kind of habitat, and they differ in size. So that enables us to look at how each species is utilizing the habitat, and how do they avoid competing with each other so strongly that they start driving one another extinct.
These species live in savanna, grasslands interspersed with small clumps of trees. At the heart of each clump is a termite mound. Termites concentrate soil nutrients and moisture, enabling the growth of palatable trees and shrubs rarely found elsewhere.
So this is forage that's just incredibly nutritious. I mean, this is cheeseburger, basically.
Ecological theory predicts that to coexist, each species must occupy a unique niche, defined by the combination of habitats and foods it uses. Rob has enlisted mammalogist Ryan Long. Together, they're using new technologies to gain unprecedented insights into the lives of these animals. To study habitat use, they need to track the movements of each species through time. Today, they're placing a satellite collar on a kudu, the largest species.
She's right here. Got it? Alright. Start. Yes. Alright, 3 minutes.
The most critical piece, obviously, to get this collar on. It will provide an hourly location for 10 months or so.
Next, the researchers need to know exactly what each species is eating. This has long been difficult, because it is often impossible to identify plants while watching an animal feed in the distance. Rob's team is pioneering a new approach called DNA metabarcoding. The researchers collect animal dung, which contains undigested plant cells. They isolate the plant DNA from those cells and match it to a reference library of possible food plants.
This is a sample about as fresh as you can possibly get it. Sometimes you get nothing at all, but we actually have nice, fully formed pellets, a very clean sample, no contamination from any kind of plant DNA,
because it came straight from the inside of the animal.
The final step, a reversal injection to wake her up. The team has collared 20 individuals of each species. Now they can start to build a picture of each animal's ecological niche by monitoring its movements and diet through time.
So what we can do when we have our GPS collared animals, we can get multiple fecal samples from them over the span of several months. Over time, what that'll enable us to do is we can actually look at the dietary profile of each individual.
And so what we hope to do with that animal is then follow up, right? Collect additional fecal samples over intervals so that, just like if someone were following you guys around, writing down what you ate for breakfast, what you ate for lunch, what you ate for dinner, what you ate for snack, doing that every day, we'd get a pretty good sense of what you like to eat. In a similar way, we're just trying to do the same thing for kudu. And that can help us answer the questions, for example, do individual kudus have dietary preferences? Like probably you guys, like some of you like Brussels sprouts, some of you hate Brussels sprouts. There's no reason really to necessarily expect that animals should be any less kind of individually variable. And that's something that we don't know about and we're going to try to figure out. But what we've done so far is use it to untangle this food web. I told you that this was impossible to do with binoculars alone, and therefore we don't know anything about it. Well, we now know a little bit more about it. So each of these lines here in this array represents a single plant species. Okay, we know it's a species, and it's color-coded by the plant family that it belongs to. And there's only two that I really want you to pay attention to here, which is the Poaceae, those are the grasses, which are obviously a hugely important plant in the food web, and the Fabaceae, and that's the pea and bean family, the legumes. Okay, and so those, and they include the acacia trees as well as a bunch of other plants that animals like to eat because they're very nitrogen rich. They fix atmospheric nitrogen and therefore make very good food. So you can see that up on the left, there's a bunch of green bars corresponding to grasses, and down on the right, there are a bunch of red bars corresponding to those legumes. And now we can look at a bunch of different species and ask exactly what they eat. So Grevy's zebra, critically endangered species that lives in this part of Kenya, something like 2,000 of them left in the world. And you see they eat almost exclusively grasses. Plain zebra, similar set of grasses, but not entirely the same. And I should say also the width of the lines here connecting indicate the frequency of utilization of these different plants. There's cattle, buffalo, so these are all grazers. Now we switch on the other side to our mixed feeders and browsers. That would include elephant, impala, and so you can see, especially with the impala, that it's eating a mixture of grasses and these red legumes, acacia trees, and other things. So it's conforming to our mixed feeder model. And our friend the dik-dik eating almost exclusively browse, very little grass. Okay? So what I just showed you is pretty and it's cool, but it's very difficult to quantitatively interpret it, right? So we need another way of doing this. And what we're going to use is something called a non-metric multidimensional scaling plot. It's a statistical ordination technique. And the key thing to keep in mind, for those of you guys who haven't encountered this before, is just that when I showed 2 points on this plot, ones that are closer together are more similar, and ones that are farther away are more different. So I can put plain zebra on this map, and you see that the points are kind of clustered, meaning that one plain zebra diet is pretty similar to another, except there is one exception out there, an outlier. I can add Grevy's zebra, and you see something interesting, which is that, again, we have an outlier up there, and again, they're clustered with each other, but they're actually overlapping but distinct from plain zebra. Okay, I can do the same thing for buffalo and cattle, and again, you see the same pattern. Like cattle eat what other cattle eat, and buffalo eat what other buffalo eat. But despite the fact that these are both two similar kinds of cow in the savanna, they have non-overlapping diets. And elephant and impala, a very similar thing is true. And finally, for dik-dik, totally off on its own. Right? So what this is saying to me is several things. First, that the grazer-browser spectrum, that classic mechanism for herbivore niche partitioning, is only part of the story. And you can see it very clearly, moving here from left to right. There's the grazers, the strictest grazers. Zebras eat like 90%, 95% or more grasses. And the buffaloes eat about buffalo and cattle eat 60% grasses, and on down to dik-dik, which eat almost no grasses. So this is the grazer-browser spectrum on the x-axis. The y-axis is a whole new dimension that we did not know about before of niche partitioning at the level of plant species. And what that means is that the Grevy's zebra and the plains zebra that eat the same amount of, same total amount of grass, 95% in their diets, are eating different subsets of grass species. So specifically, there are 17 species that are not shared between the two, that either only plains zebra or only Grevy's zebra will eat. So this is, what the previous understanding was, as is often the case, was not wrong, but it was incomplete. And now we are completing that with the aid of new techniques, right?